Hi, my name is Andrew Johnston. I'm the invertebrate collections manager for the Neon Biorepository at Arizona State University. And I'm here today to talk to you about Symbiota. Symbiota is an open source biodiversity data management software. Here's its website. And what really makes Symbiota kind of stand out is that it's designed to manage portals. So there are at least 54 Symbiota portals that are currently online and available to the public. And within those portals, at least 880 collections are live managed. Their primary source of collections data management happens within Symbiota, but more than 1,700 collections contribute their data to these portals, which gets close to 90 million occurrence records and 42 million images that are mobilized through Symbiota. And Symbiota is open source. It's available here on GitHub. You could download this, put it on a server, install your own instance if that's what you're interested in doing. But it's designed to make these community portals. So I'm going to walk through a number of portals that currently exist and use the different portals to highlight different features which are actually available to basically any Symbiota portal. But here's Ecdysis. This is a smaller portal which is meant for collections who are live managing their invertebrate arthropod data within a Symbiota portal. So we can go to search here and see all of the collections that are contributing data to this portal. So the nice thing is each one of these collections doesn't need to have their own in-house database system or server to manage the data on. They can join this community effort. And so we have institutional collections here, and we also have individual, maybe graduate students and other project-based collections that are managing their data here. So as an example, we can go here to the Arizona State University Hasbrook Insect Collection, where we can see that the team there has managed to digitize about 175,000 records from the collection. Because I'm logged in and I have permissions, I can toggle some tools here. So these are the tools that we would use to manage data within this collection. So we could add new occurrence records, we could edit existing records, we could manage loans that we're sending out to other institutions. We can also do things like manage who can see rare species if we want to try to limit who can see some of these occurrence records for rare species, and batch upload and other things. But to show out just a little bit of what this might look like if you're managing data live here. So here's a table of ASU specimens, but we could query this, perhaps say our family is Apidae, and we could display a table and see some 5,000 records here of apids. And we can look at them as a table, or we could go to a particular occurrence record. And this is what internally our records look like when you're editing them. Metadata about the specimens, the identifications, the locality and collecting events, metadata maybe about the habitat, and other metadata about the specimen in general. And we could go and see the determination history of this specimen. It seems that there's only one determination event here, so there's not a history. Load images and store those here. We could also link this to other resources online, perhaps a BOLD or GenBank sequence record. We could also code traits for this individual within the portal. And as an admin, we can look at things like edit history and how this specimen is connected to other things within the portal. So that's kind of a very quick look at the back-end management, but we can immediately go and see the public record. So one nice thing about Symbiota portals is as soon as you commit the data to the database, the data are available to the public, and you can come here and see all of the data that was input for this specimen. So I know that we'll see some other presentations about SCAN here in our workshop. SCAN is also built upon the Symbiota software. And here I just want to show the power of larger community portals, right? So here we see many more collections. So some of these collections manage their data live on SCAN, but most of them are snapshots. These are data that are contributed from the collection from their internal database or management program and put here on the portal. So here we could maybe search for Apis mellifera and get a large number of results, 244,000 of them, where we could download them, map them, et cetera. So that's how these collections kind of come together within a portal, and you can see that. A third portal is the Bee Library. So this is a portal that was created for the Big Bee TCN. And here we can go and search our collections, and we'll see that it's a smaller number, and this is really focused on bee records. So there are some contributing collections within the TCN, but also some external data providers. Again, we can, as an example, just look at our ASU collection here, and we see that there's only 13,000 records in this snapshot. So our data, again, are stored live on Ecdysis, but these portals can talk to each other, and we can serve over just our bee records as a snapshot to this taxonomically focused portal. One thing I want to highlight with this portal is we can look at checklists. Now, I haven't made any of these, I'm just showing off other people's works, but as an example here is the UC Santa Barbara part of their campus, I believe, a checklist created by Katja Seltman, 26 bee species that have been recorded from there so far. But we can see not only are there names of these bee species there, but they are validated, substantiated by voucher occurrences within the portal. So here is the voucher that is linked to that name. You could look at this picture or request a loan of that specimen if you wanted to kind of drill down on whether that species is actually there. You could also come over here, instead of seeing the notes and vouchers for this, we can display it as images, and we can build upon all the images in the database, and now we can get kind of a visual representation of the bee diversity from this site. And I'm sure we'll hear about some more things that are happening here in the bee library and other times in this workshop. So finally, I want to mention just the NEON Biorepository portal. So NEON is the National Ecological Observatory Network. It's an NSF-funded project where ecological and organismal data are collected at these 81 sites across the US. And physical samples are sent to us at the NEON Biorepository, where we have another Symbiota portal that we manage all of our data in. And I just want to use this to highlight some other types of things that and data that can be managed in Symbiota portals. So here is a specimen, again, a public record. We have a lot of metadata here about the habitat and description of where this is. We see that this specimen is actually on loan. So I mentioned loan management here. We can see some images. So this 1.5 millimeter critter we found, we can come here and see the identification history. I was pretty sure it was a hymenopteran at first. And then I drilled down a little further and found, oh yeah, it's this kind of really cool genus Basona. It's a braconid wasp. And it turns out it's undescribed, and so that's why it's on loan. But not only can you see the termination history, but we can also see that this occurrence has a relationship, a link to another record. So this occurrence here, this individual specimen, is a subsample of this record. And this record is a bulk sample. We have a 50 mil tube of pitfall bycatch, which NEON has generated, and that sample is sitting in our collections. And that sample here in our portal is linked to 4 pinned individuals. So you could imagine a bee monitoring project where you have your bulk samples and all of your specimens being linked to that. All of these data are served along with the occurrences to GBIF and other aggregators from this portal. Another sample I want to show, this is a mammal, a little Paramiscus mouse. We have some images here of the specimen. Again, a lot of metadata, including some physical characteristics that were collected by the team here that prepped these. But here we have material samples. And this is something that the entomological community hasn't quite started using as much as some others have, but we see that there's a study skin, a skull, a postcranial skeleton. The preparators have pulled tissue samples, kidney, heart, lung, liver, GI tract, including an embryo from within this animal. And all of these tissues are then stored perhaps in ethanol. Some of these are stored in liquid nitrogen in our cryo collections, and some at -80 in our freezer farm. So all of these are, you could think of them as subsamples, but they're really kind of these material samples that are related to this parent organism. So you could think of dissections and DNA extractions and other preparations within our collections, and how all of those metadata can be linked and used within a portal and shared across other portals. And finally, I want to show one really cool thing as thinking about monitoring biodiversity. So this is a checklist. Again, we saw some actual bees. This happens to be carabid ground beetles, and checklists can be hierarchical. So this is for the desert Southwest carabidae, which is built off of a smaller site in Arizona and one in New Mexico that together are hierarchical to form this. So again, if you're monitoring across large space, you could have a hierarchical sampling structure and create checklists at these different levels. We have a species list here, just like before, but here I'm going to click on this little key symbol and open up an interactive multi-entry key. Just like before, I can display this as images, see all of the taxa that are included here. And again, think of these maybe with B diagnostic characters. I know that there's 2 superorbital CD on a specimen I have in front of me. Well, now I've narrowed it down by a little over half. Maybe I'm not quite sure about these characters, and I can see, oh yeah, this is what the plica looks like on the elytron. Yeah, that's present. Oh, well, that got me down to 3 specimens. And here now we have some new characters that are showing up for this subset of taxa to help us discriminate between them. And maybe my labial palpomere 2 is pleurocetos. Perhaps this is what I have. I can look at it, compare it with images in the portal, and help to identify stuff. So these are just some of the tools that are available in these portals, and these portals all can talk together and coexist within a broader biodiversity data network. And I just want to quickly close. Symbiota.org is a great resource. There's a lot of documentation and help there. Also, for contact, help@symbiota.org will get to the team that manages and develops Symbiota, and they are wonderful. I'm here to talk about this, of course, too, but I especially just want to shout out this Symbiota Support Hub team that's full of biodiversity informaticians, developers, data managers, project managers, and they all are here at Arizona State University, and they're here to serve the collections community. And with that, just thank you for listening, and I look forward to interacting with you all here at this workshop. Thank you.